My name is Robert Hooke, and I am from the year 6055. I hope this recording has reached to you soon. Last night I was in my basement and I discovered a very astonishing thing. I discovered cells. I discovered that the smallest living thing that ever can happen is cells. All living things are made up of cells. Cells are the smallest unit of life. New cells only come from pre-existing cells. That is my discovery. I hope you receive this message. Well, thank you, Mr. Hook. That message was received and summarized what Mr. Hook was just saying right now. And Mr. Hook was saying that there are three. Cell theory has three things inside it. The first, that all living things are made up of cells. The second, cells are the smallest unit of life. The third, new cells come from pre-existing cells. And that's pretty obvious. There are two kinds of cells. We have a prokaryote, we have eukaryotes. And really, they have some differences. And the differences is that the prokaryotes don't have any membrane-bound organelles. And this includes bacteria, cyanobacteria, archaea. And eukaryote includes more sophisticated cell organisms. And we'll talk about that later in a different video. But both present basic biochemical processes, and this includes that they include photosynthesis, they are going through oxidative phosphorylation, the use or to produce the ATP, or to use ATP in the Krebs cycle. And these are certain processes that both of them are both of them possess, but eukaryotes have membrane-bound organelles, we know that, and they have mitochondria, lysosomes, and nucleuses, Golgi apparatuses, and a lot more that prokaryotes don't have actually. So this video is dedicated to prokaryotes, so we're going to stick on to prokaryotes. Taxonomy refers to the classification of living organisms, and the highest division in the taxonomic cycle, or the taxonomic divisions, is domain. There are three domains, and the three domains are bacteria, archaea, and eukarya. If we break it down even further, we will figure out that the smaller subunit at this point will be kingdoms. And eukarya has three kingdoms. We have Animalia, Plantae, Fungi. But bacteria and archaea include bacteria. So you might be a little bit confused, saying, hey, why do we have... people get this mixed up often in the domain. And this was, if you're taking the early on classes in biology, you will definitely get this on your test. The name the three domains, and that's bacteria, archaea, and eukarya. So let's get into the structure of the bacteria. So the bacterial structure and classification. So the cytoplasmic content of the bacteria does not include any internal membranes. It has a circular chromosome, and it has polycistronic translation. Polycistronic, remember, refers to the fact that when translation of a certain transcript, which is the mRNA, happens, we have more than one gene being translated, and that means more than one protein being translated. And this can also happen through polyribosomes. Polyribosomes refers to the fact that one transcript can have multiple, or one mRNA has multiple ribosomes attached onto it that's translating at the same time. We have something called plasmids also inside bacteria, and plasmids are extra chromosomal genetic elements. And let's say that we have a cell over here, and this is its normal genetic information, and then we notice that there is something else over here. That's the plasmid, as well as green, because green is the ideal color for the plasmid. Well, their size is not relative. Let's get a relative size. The plasmid over here is a genetic element. It's an extra chromosomal genetic element. And this plasmid is a source of exchanging and passing on information from one bacteria to the next. So it's useful in that sense. So plasmids are advantageous. They're useful genes, and new abilities can be passed on, and they're autonomous, which means that they can replicate on their own. So many copies can be made inside a cell and exchanged with another, and this process is called conjugation. There are certain shapes of bacteria. So bacteria can have three possible shapes, as far as you've noticed it. So it can be coccus, which means that it's circular. It's a circular bacterial shape. Or we can have bacillus, which is a structure which is like a rod. Well, it's like structure like this over here. And then we have spirilla, which is a spiral structure. The bacillus is a rod-shaped structure in a sense. And we have bacteria. So bacterial cell membranes consist of I always get this wrong, peptoglycan. So peptoglycan is essentially cross-linked chains between the sugars and the amino acids. So it's just this. We'll get into more detail of the peptidoglycan. We'll get into detail later on. But the important point to understand at the moment is that the peptidoglycan is essentially responsible for the cell membrane strength. And if we have antibodies that are given to us when we are sick in combat to bacteria, or we have lysozymes, they are targeting the peptidoglycan. And the peptidoglycan will eventually be destroyed because of the enzymes. And this leads to the protoblast, which is an osmotically fragile cell wall. And this can cause lysis to occur, which is the entire cell just dies that way. But if you want to memorize protoblast, just think of this. Break this word down. Look at that. That's 2 is not copied over here. To blast. It's protoblast. It's protoblast. It's going to blast. It's osmotically fragile. So hopefully this video made sense, and we're going to continue on to talking about gram staining and the positive and negative membranes possible, and that's a reference to the peptidoglycan.